This is part 2 of 2 on chapter 8, Microbial Metabolism. In the first part of this chapter, we looked at enzymes and what enzymes do. To review, enzymes speed up chemical reactions so that cells can do things faster. And they do that by lowering that activation energy, that energy needed to get things going. So in this part of the chapter, we're going to look at cell metabolism, so looking at energy a little bit, and then we're going to look at cellular respiration and photosynthesis. And those are the two big metabolic reactions that we find in cells. So first off, we're going to look at energy. So all living things, they need a source of energy. And energy is defined as the capacity to do work or to cause change. And there's lots of different forms of energy. There's thermal energy, which is heat, radiant energy, electrical energy that we use to power things, mechanical energy, atomic energy, and chemical energy. In organisms, we get energy in the form of chemicals. So either photosynthetic organisms, they have to get energy from the sun, they use that to make chemicals, and then they break those different compounds down. Or if you think about us as humans, we need to eat food in order to get energy. So we eat all of these molecules, we get them from our food, we break those down, and we get energy from the food. So in cells, cells need energy. That's one of the characteristics of life. So cells manage energy in that chemical form, in that chemical reactions form that I was just talking about. So there's different types of reactions. There's endergonic reactions, that's the first one up here. Endergonic reactions, they consume energy. So these reactions, we have to use energy to actually get them. So here we have energy, you add a molecule A plus molecule B, you have an enzyme that does that reaction, because every step needs its own enzyme. That enzyme helps to speed up the reaction, it helps to facilitate the reaction, and then you get out product C at the end. So remember, endergonic consumes energy. Exergonic reactions, they release energy. So think of X or exiting, the energy is going to exit out. So in this instance, we take molecule X and Y, and of course we need another enzyme to do this reaction to help speed it up, to help facilitate it. And then we get molecule Z plus energy that's released. This energy that can be released from these exergonic reactions, the cells can store it in what are called high energy phosphate molecules. So any energy that's released from an exergonic reaction, we can store it for a little bit, and then we can use it to do an endergonic reaction. So it's called coupling of the two reactions. This graph is showing an exergonic reaction. So on the x-axis we have time, so left is starting, right is finishing the reaction. Then on our y-axis, so the up and down, it's the energy level in a chemical compound. So remember, living things, they get energy by breaking down chemicals, or large molecules that we get from our food. So if you look on the left, upper left-hand corner, we have glucose, which is a sugar, it's a carbohydrate right here. And this glucose molecule is going to go through a process called cellular respiration. And as cellular respiration occurs, we're going to break down that glucose molecule, and we're going to extract energy from it. And I'm going to show you how this actually works, but for right now, you get energy in the form of hydrogen ions that have electrons on them. And then we store that energy into ATP, which is that yellow molecule by all the blue arrows. And then that ATP is going to be used to perform cellular work. So ATP is that high energy phosphate molecule that's storing this energy that we just captured. And then, of course, we also get end products. In cellular respiration, the end products are water and carbon dioxide. So those are kind of the waste products. So we breathe out carbon dioxide to do this process. So this is just showing how we extract that chemical energy from glucose molecule from food, and we store it into those energy intermediates like the ATP. So this reaction is actually called an oxidation reduction reaction or a redox reaction. And redox reactions always occur in pairs. Okay, so if you look on the left, we have our glucose molecule. We also need oxygen as well. So what's going to happen to the glucose is going to be oxidized. And if you look, glucose is C6H12O6. And to oxidize something, it means you're just going to take hydrogens off of it. So you can see that. So we go from a molecule that has carbon, hydrogen, and oxygen. We take those hydrogens off. We end up with carbon dioxide, which contains carbon and oxygen. So that's oxidation. Then those hydrogens, we're going to attach them to oxygen gas. So when you attach hydrogens to something, it's called reduction. So we take our oxygen gas on the left, we add hydrogens to it, and we end up with water over on the right-hand side. So again, oxidation, you take hydrogens off. Reduction, you're adding hydrogens on. And then as we do this exchange of hydrogens, we're going to get energy out of it. And that's going to be stored in that ATP that we just looked at. So ATP, if we look at it a little closer, ATP is called a metabolic currency. So it's the currency, it's kind of like when you use money to buy stuff, ATP is used to do things in the cell. So ATP, what it looks like, so it's an adenine nucleotide. So if you remember from DNA, if it's actually a RNA nucleotide, so it has a ribose sugar, has adenine as the nitrogen base, and then instead of just one phosphate group, it has 3 phosphates. So it's triphosphate, 3-phosphate molecule. So these, usually we just have one phosphate attached to a nucleotide, if you remember that from the DNA. So these other 2 phosphate groups that are attached, they contain a lot of energy. So if you remove these terminal phosphate groups, so the one on the end or the second one, that's going to release energy when those come off. So this ATP is utilized and replenished in a constant cycle. So we're constantly taking these phosphates off to release energy, and then we put phosphates on to store energy. So ATP is constantly being created and destroyed, and it's just used to exchange energy from one form to another. There are 3 ways that ATP can be formed. So these are the 3 different mechanisms. So the first one is called substrate level phosphorylation. This is where we transfer a phosphate group from a phosphorylated compound directly onto an ADP. So an ADP has 2 phosphates, so we want to take a phosphate off some other molecule, put it on to get adenine triphosphate, or 3 phosphates. So, and this is the image shown here. So we have a phosphorylated substrate, so we have some molecule that has a phosphate group attached to it. We're going to take that phosphate off of that substrate, that green molecule. We're going to put that phosphate onto an ADP, which only has 2 phosphates. So if you have a molecule of 2 phosphates, we add on another one, you get adenine triphosphate, or 3 phosphates. And then that substrate is just normal, back to the green molecule. In substrate level phosphorylation, the important part of this is it uses an enzyme to do this process. So the enzyme, remember, is a protein. It helps to speed up this reaction, and it helps to take that phosphate off the substrate and put it onto the ADP to make your ATP. Other types of formation of ATP, you can have oxidative phosphorylation. This is where you have a series of redox reactions occurring during a respiratory pathway. So we're going to see this in cellular respiration. The third type is photophosphorylation. This is when ATP is formed using energy from the sunlight. So we'll get more into these other 2 types later on. These other 2 types, they do not use enzymes to create the ATP. So pathways of bioenergetics. Bioenergetics is just a study of the mechanisms of cellular energy release. It includes catabolic and anabolic reactions. So we're going to be breaking things down and building them up. And the two important processes we're going to look at are called cellular respiration and photosynthesis. So first off, we're going to look at cellular respiration. And there's three different kind of things that can happen during cellular respiration. So we have aerobic respiration. Aerobic means with oxygen. So aerobic respiration is when you have oxygen present. And this is where your glucose molecule is going to go through glycolysis, the Krebs cycle, and the respiratory chain. So it goes through 3 steps where we're going to break down that glucose molecule to get energy. We also have anaerobic respiration, which is without oxygen. So anaerobic respiration, you can also have glycolysis, you can have the Krebs cycle and the respiratory chain, so you still have the 3 steps of cellular respiration, but instead of oxygen being used at the very end of the whole process, there's going to be some other type of molecule that's going to catch the final electron acceptor. And this will make more sense when we get into there. But basically, aerobic and anaerobic, you have the same steps, but aerobic, you have oxygen at the very end. Anaerobic, you have something other than oxygen. And then the third strategy is called fermentation. This is in an anaerobic environment, so without oxygen. And fermentation just is the process of glycolysis. And then fermentation also produces different types of organic compounds that we can use to identify different bacteria species. Okay, so this is the overview that you have in your homework. So on the left-hand side, this is showing the aerobic respiration with oxygen. So you have glycolysis, which is in the orangish box. You have the Krebs cycle, which is the green cycle in the middle. And then the final part, the electron transport chain at the end. So this first panel on the left, again, is aerobic respiration with oxygen. And you can see that at the very end, so oxygen is the final electron acceptor, because you have oxygen available. Also, at the very end, the maximum amount of ATP that's produced is 38 molecules of ATP. So that's important when we look at the other two. In the middle, you have anaerobic respiration. So you still have glycolysis in the orange box, the Krebs cycle in the green circle, and then your electron transport chain at the end. And then remember, anaerobic is without oxygen. So at the very end, you can use different molecules other than oxygen. And the maximum amount of ATP produced, it ranges from 2 to 36. So you can see anaerobic respiration, it's not as efficient as aerobic respiration, but you still get quite a few molecules of ATP produced. And then on the very right, this is fermentation. So in fermentation, it just has glycolysis in that orange box. And then those pyruvates that come out of glycolysis, you can see pyruvate, they have to go through fermentation. And this is where different types of alcohols or organic molecules are produced in the pinkish purple box. So an organic molecule is the final electron acceptor. One of these would be the production of lactic acid, or another important one that's used a lot is the production of ethanol, or just alcohol. Fermentation is even less efficient compared to the anaerobic and aerobic respiration, so we only get 2 ATP that are produced. That's the maximum. So fermentation, it's not really that great, but we get some energy out of this process. Okay, so in aerobic respiration, and again that's with oxygen, here's what actually happens. So you have glycolysis, which is first. That's that orange box. So you take a glucose molecule, which has 6 carbons in it, and you basically split it in half. So you end up with 2 molecules of pyruvic acid, which each have 3 carbons. So you're just splitting glucose in half into a pyruvic acid molecule. We get NADH that's generated. NADH is holding hydrogens. So we're ripping hydrogens off of the glucose and putting them onto this NADH molecule. In addition, we get 2 ATP, so 2 energy molecules are created during glycolysis. That pyruvic acid then goes into the Krebs cycle, which is that green circle. The Krebs cycle, it's a metabolic cycle, so things go into it, things come out of it, like we looked at in the previous section in this chapter. So this Krebs cycle, it's going to process that pyruvic acid. And while it's processing it, we're going to generate 3 molecules of carbon dioxide, which are released as byproducts. We get more NADH, so we're taking off more hydrogens and storing them on this NADH. And then we can also store more hydrogens on a molecule called FADH2. So again, those are just storing hydrogens for now. We're going to use those later. And the Krebs cycle also produces 2 more ATP, so we have 4 total coming out of this. So aerobic respiration, it produces a maximum of 38 ATP, and so far we only have 4 that have been produced. So the rest of the ATP molecules are produced during the last step called the electron transport chain. So electron transport chain, it accepts the electrons from your NADH and FADH2, so it's going to rip hydrogens off of these molecules, and we're going to use those hydrogens to create 34 additional ATP through a process called oxidative phosphorylation. And remember, that's one of those 3 ways to make ATP. This slide is showing the fate of pyruvate. So we just looked at glycolysis, so we take that glucose molecule that has 6 carbons, and we break it in half into 2 pyruvic acid molecules that have 3 carbons. So if we go down the middle of this picture right here, if you have oxygen available, your pyruvic acid is going to go through the rest of the cellular respiration. So it goes in the Krebs cycle, and then you have the electron transport chain at the very end. Sometimes pyruvate can not enter into the Krebs cycle, and that's where you have the fermentation. That's where you're going to get all those different organic molecules or alcohols that are produced. So after glycolysis, after you take your glucose molecule, you divide it into half into the 2 pyruvates. Those pyruvates are then going to enter into the Krebs cycle. And the Krebs cycle, this is the actual Krebs cycle. You don't have to memorize it in this class, but just know that the Krebs cycle is also called the citric acid cycle, because citric acid is the first product that's produced. So it actually starts on the left-hand side. Your pyruvate enters into the cycle, and then it's processed. There's 8 reactions. You can see that they're numbered 1 through 8. Each of those reactions has its own enzyme. So for example, if you look at reaction number 1 on the far left. So reaction number 1, you take oxaloacetate molecule. You're going to attach acetyl-CoA using the citrate synthase enzyme to create citrate. So you can see any of the words that end in ACE, the ASE, those are enzymes. So you can see there's enzymes for each step. And then we also have the reactants and the products for each step. And again, you don't have to memorize them, but just notice that along the cycle you can have NADH being produced, you have carbon dioxide being produced, you have ATP that's coming out of the cycle. Down at the bottom, step 6, you have FADH2 that's being produced. Step 8, we get more NADH. So these products come off at different steps within this cycle. And then the last thing about the cycle is it is a cycle, so things come into the cycle, things come out of the cycle, but you always end up at the beginning. Then the final part of cellular respiration is the electron transport and the oxidative phosphorylation. So this is that final processing of your electrons and hydrogens that are on your FADH2s and NADHs, and we're going to use those to create ATP. And this is process starts with the electron transport chain, or ETC. So your electron transport chain, it's going to shuttle electrons down a chain of molecules in a membrane, and that causes energy to be released and then subsequently captured and used by an enzyme called ATP synthase. ATP synthase, it's an enzyme complex that's used to produce ATP. And you can kind of see by the name of the enzyme, so synthase means to create, so it's creating ATP. And that process is called oxidative phosphorylation. So here's an actual image of your electron transport chain. So if you look on the left-hand side, you can see we have all these different proteins embedded into a membrane. And those proteins, like the purple, orange, the green, the pink, the red ones, right here. So the first protein, called NADH dehydrogenase, that enzyme is going to dehydrogenate. It's going to take hydrogens off of your NADH. So you can see we have an NADH molecule, goes into the NADH dehydrogenase, so a hydrogen is ripped off of it. That hydrogen is then pumped up to the top of your membrane up here. We also get an electron that's ripped off. That electron travels down these different colored proteins. As your electron travels down, it causes hydrogens to be pumped to the top of the membrane, up at the top here. So we're going to end up with lots of hydrogens at the top of the membrane, no hydrogens at the bottom of the membrane. And things want to go from high concentration to low concentration. So these hydrogens want to go from the top, where they're really concentrated together. They want to move down to the bottom so they can spread out and get further away from each other. And since they're ions, in order for them to actually do this, they have to go through ATP synthase over on the right-hand side. So ATP synthase, like I said, it's an enzyme that synthesizes ATP. So it's going to allow these hydrogens to go from the top to the bottom, and as it does that, it's going to create an ATP molecule down at the bottom here. So this is that oxidative phosphorylation. So we have a hydrogen gradient set up. Those hydrogens want to go across the membrane. As they go across the membrane, they're used to create our ATP. In eukaryotic cells, this process occurs in the mitochondria. And this is shown here. So in the mitochondria, remember, mitochondria is responsible for cellular respiration, which you just learned in the previous unit, hopefully. So in the mitochondria, there is an inner membrane that has lots of surface area, so it's very folded. And so that's the membrane that's used during this formation of ATP. So if you look at the picture on the left-hand side, in the very inside, where it's kind of light blue, you don't have very many hydrogens, you don't have very many of those yellow circles. But in between that inner membrane and the outer membrane, you see lots of yellow circles. Those are hydrogen ions. So you can see we have this gradient set up, lots of hydrogen ions on the outside, very few on the inside. So those hydrogens want to go from high to low concentration, and again, to do that, they have to go through ATP synthase. And then as they go through the ATP synthase, ATP is synthesized. In prokaryotic cells, since prokaryotic cells don't have mitochondria, they do this using their cellular membrane here and their cell wall. So it's kind of the same concept, but in between the cell membrane and the cell wall of prokaryotic cells, that's where that hydrogen gradient is going to be set up. So in the picture, the cell wall is the dark brown structure. Your membrane you can see as well. So those hydrogens, they're pumped into that space between the cell wall and the cell membrane. So lots of hydrogens in between. Those hydrogens want to get back into the cell where the low concentration is. And again, same concept, to get back into the cell, they go through ATP synthase. As they go through, ATP is synthesized. Okay, so as this process is occurring, we're going to have electrons that are just being created. So these electrons at the end of the electron transport chain, they have to be caught. And this is called the terminal step. So this is where oxygen comes into play. If oxygen is present, oxygen's going to catch those two electrons, going to catch those electrons that are coming off the end of the electron transport chain. It's also going to take up 2 hydrogen ions from that solution. And using that, we're going to get water that's formed. So in aerobic respiration with oxygen, oxygen's going to be that final electron acceptor at the very end of this whole process. So you have hydrogens, electrons, and oxygens, and you end up with water that's produced. Okay, so this is showing that theoretical ATP yield for aerobic respiration, which is 38. So we got 2 ATPs from glycolysis, 2 ATPs from the Krebs cycle, and then the remainder, the 34 other that we need, come from the electron transport chain. And you can add them up over on the right hand side to get up to the 38. Okay, so next we have anaerobic respiration is without oxygen. So it's basically the same thing that we just went through. Functions like the aerobic respiration, we still have glycolysis, the Krebs cycle, the electron transport chain, except at the very end, that final electron acceptor, instead of oxygen catching that final electron, we have other ions that will catch it. So things like nitrate or nitrite are good examples. So most obligate anaerobes, so if you remember from chapter 7, we talked about obligate anaerobes. Those are organisms that have to live in an anaerobic environment. So if they have any oxygen available to them, it will kill them. So these obligate anaerobes, they use the hydrogen gradient generated during glycolysis and the Krebs cycle, and they use something else other than oxygen at the very end. So obligate anaerobes are good examples of organisms that use the anaerobic respiration. And then our third strategy is fermentation. And remember, this just has that glycolysis process. So we have incomplete oxidation of glucose or any other type of food that the cell wants to break down, and no oxygen available. So your glucose is broken down into 2 molecules of pyruvate. That pyruvate has to be converted into some type of organic compound to be finalized. And we'll look at different ones, but lactic acid is what we make when we go into fermentation if we have a deficit of oxygen. Or things like bacteria and fungi, they produce ethanol or alcohol that's found in beer and wine. So this fermentation process, it produces a small amount of ATP. It only produces 2 ATP because it only does that glycolysis process. So not a very efficient strategy, but you still get ATP, so those cells can still live and do their metabolism. So this is showing more of like the chemistry thing of it, but basically you have glucose, goes through glycolysis, like I just said, and that pyruvic acid can be converted into different alcoholic compounds or different acidic compounds. And this is showing all the different products that can be created from this pyruvate fermentation. And these products, you can actually use them to identify different bacteria species. So if your pyruvate gets converted into ethanol, you probably have a yeast molecule that you're growing. If the pyruvate gets converted into, let's go to the bottom left, so the proponic acid, you probably have a proponibacterium. So you have a specific type of bacteria. If your pyruvate gets converted into carbon dioxide and hydrogen, it's probably a Proteus species, so that's going up towards the right over here. So you can actually use these different products that are produced to identify different species of bacteria. So pretty cool, you can do that in lab. So we looked at cellular respiration. The other big process is photosynthesis. And this is actually Earth's lifeline. So in order to actually get food or to get chemicals that we can eat and break down to get energy, we have to make those chemicals. So the ultimate source of all chemical energy in cells comes from the sun. Yay for sun. So this photosynthetic process, I really like photosynthesis, probably because it's something that my own body can't do, so I find it more interesting, which might sound kind of weird, but anyways, love photosynthesis. So we take carbon dioxide and water, so you're taking two inorganic compounds, sunlight, and you use that to create glucose. So you take inorganic molecules to create an organic molecule. Like, how cool is that? So that glucose then can be turned into different types of food, and then they also produce oxygen, which we need in order to live. So these photosynthetic organisms, pretty awesome. So photosynthesis, it occurs in 2 stages. That's another reason why I like it. It's a little simpler, less stages to it. So there's a light-dependent reaction, which depends on light. This is where we have our photosynthetic organisms, like the plants and the algae and some bacteria, the cyanobacteria. So that sunlight, it comes in, hits those plants or bacteria or algae. They absorb photons of light using chlorophyll, which is that green pigment. They also use other types of pigments, so carotenoids or phycobilin. I think that's how you pronounce it. But you have these different pigments that absorb basically color, light. And this photosynthetic reaction, again, I'm not going to go into a lot of detail of it. I do that in principles of biology. But the sunlight, it comes in, it hits these chlorophyll, these pigments, the carotenoids, and it converts that sunlight energy into ATP energy. So we're just capturing sunlight energy and turning it into ATP energy that can be used by the cell. So once we have our ATP that's created, then we have our light-independent reaction, or it's called the dark reaction. So this is the part that doesn't need the light. It's also called the Kelvin cycle. So we're going to use that ATP that we just created in the light-dependent reaction. We're going to use it to take our carbon dioxide. We're going to fix or attach that carbon dioxide to a molecule called RUBP, or ribulose 1,5-biphosphate. It's RUBP. So we take carbon dioxide, attach it to the RUBP, and then it goes through this Kelvin cycle, and out of our Kelvin cycle we get our glucose. And we also get oxygen from our light-dependent reaction. So this is the Kelvin cycle. It's another metabolic cycle where things come in and they exit out. So let's see. So way up at the top we have our little carbon dioxide molecule. It's attached to our RUBP to create a 6-carbon molecule. We then split that 6-carbon molecule into 2 3-carbon molecules. We add in ATP, we add in energy, and then out at the bottom we get glucose, or sugar, that's produced. So that is basically photosynthesis, very quickly. Very cool process, but we don't have enough time to go into a lot of detail, so you just basically need to know kind of the main points of these cellular respiration and photosynthesis, so kind of where it happens, how many ATP are produced, or what's produced, what goes into it, more kind of the general things about it. So this whole chapter was on metabolism, so all those chemical and physical workings in a cell. Specifically, we looked at enzymes, which are used to do all these reactions to help speed them up. And then we looked at the 3 types of cellular respiration, and then also photosynthesis.